Good afternoon. Can everybody hear me? Yes. Okay, great. Hi, good afternoon, and welcome to the Immunotherapeutics Committee meeting for the fall 2023 SWOG meeting. My name is Katie Politi, and I co-chair this committee with Siwen Huliskoven, who is here on the podium as well. Welcome to those of you who are here in person, and welcome to those who are joining us virtually on Zoom. As you can see, we have our part of the Immunotherapeutics Committee team includes our protocol project manager, Chris Kippola, who is here in the front row, our executive officer, Anne Chang, who's also here. Thank you, Anne. And our clinical trial program manager, Laura Gildner. So this is the team. Let me see. Okay, this is our agenda for today. We have four very exciting presentations, and hopefully we'll also be able to have quite a bit of discussion as we go through the different presentations. We're going to give an update on our iMatch pilot. Then we're also going to have a presentation on an effort initiated by the Society for the Immunotherapy of Cancer to really collect data on resistance, and you'll hear about sort of definitions of immunotherapy resistance and how these match what we're seeing in the real world and in clinical trials. Another presentation on navigating clinical data resources for IO studies, which matches really nicely with the presentation that we're going to hear from Dr. Twabi on the resistance data collection effort. And then we're also going to have a presentation from Dr. Marlene Koch from the Netherlands Cancer Institute on immunotherapy in breast cancer. So just with the next slide, just a couple. If you could click through a few times, please. There we go. Thank you. Just a couple of things about the Immunotherapy Committee. For those of you who are new to the committee, this is a committee that became a full research committee in 2022, last year. Prior to that, it was an Immunotherapy Research Support Committee. One of the main efforts that we have undertaken as a committee is the development of the Immunomatch clinical study with the NCI, and in particular, most recently, the development of a pilot study that you'll hear about today during the first talk from Dr. Swasicki. And one of the main objectives of the Immunotherapy Committee is really to facilitate immunotherapy trial design and inclusion and analysis of translational medicine in studies across diseases. So that's one of the objectives of this committee, is really to function across different diseases. And so one of the things that we've done is we've been gathering disease committee representatives with IO expertise who work and are part of our immunotherapy working group. And if there's some members of our IO committee working group, please go ahead and raise your hands in the room. Thank you very much for your efforts and working with us. And one of the things that we do is we hold monthly SWOG IO committee meetings to discuss any trials or concepts or anything that is coming up. And if anybody has ideas and wants input, the idea is to really be able to leverage the expertise of this group to help move those concepts forward. Next slide, please. And this really is a schematic to show you how the SWOG IO committee works. We have the committee that has the co-chairs, works with program managers, with statistics, with executive officers. Then it includes an IO expert panel, our working group that really interfaces with the different SWOG disease committees, and SWOG and IO committee members at large as well. And we work together then also with IO committees in other groups, and then with other collaborators and groups like Jackson Labs, Cold Spring Harbor Labs, or SWOG Biorepository, SWOG Stat Center, and SWOG Leadership to really advance collaborations that are disease agnostic across different committees and develop trials, IO trials, and translational medicine. And so I think that's the last slide. Next. Oh, just a reminder that we have monthly IO committee meetings on the first Friday of each month at 2:30 p.m. Central Time. And this is really, these are meetings in which we discuss initiatives and provide updates on ongoing activities. And investigators are welcome to present trials and ideas for feedback at these meetings. So all they have to do is just email us, and then we can put you on the list for those meetings on the agenda for one of the next meetings. And we really welcome people to join and attend those. So as a follow-up to these intro slides, I'd like to introduce our first speaker today. And our first speaker is Dr. Paul Swasicki, who is a head and neck medical oncologist and from the University of Michigan. And he's going to be giving you an update on our IMatch pilot study S2101. Thank you very much.
Thank you very much. It's fun to see people are larger than 1 inch on the Zoom boxes. I haven't been to SWOG actually since pre-COVID, so it's great. Thank you. And I'm presenting this on behalf of the entire S2101 group. This has been a huge endeavor and continues to be on a daily basis, as you'll hear about. And although I'm here, everyone really deserves the credit. So dating back now to some time, the goal of this pilot has actually been to explore the feasibility of the IMatch concept in a prospective trial. Although we all think of Match being this demonstrated platform that subsequently spawned Taper and many others, looking at an immunomatch is really a completely different beast using different assays and completely different workflows. So to examine the feasibility of a prospective biomarker stratification type design, as well as to streamline a central protocol operational structure, were all very key aspects before opening a large trial. As a background, we hypothesized that a VEGF plus VEGF-based therapy plus immunotherapy would reverse immune suppressive microenvironment and synergize with immunotherapy to improve anti-tumor efficacy as well as patient outcomes. Cabo plus cabozantinib plus nivolumab has been FDA approved in renal cell carcinoma, and there's activity of VEGF plus PD-1 across many tumor types. In melanoma and head and neck cancer, we'll talk about the data here, but we're prospectively stratifying patients based on biomarkers and studying mechanism of response through translational studies, as well as resistance in in patients treated this combination in distinct biological subgroups. To the right here, we can show a quick schematic showing the mechanism of VEGF inhibition plus PD-1. This is through a variety of mechanisms, some of which is still under discussion, and a little bit different tumor type to tumor type. Being a head and neck medical oncologist, a little bit lonely at SWOG. It's one mechanism through Treg depletion, as well as improvement in TIL infiltration. And those are thought to be a couple of the main mechanisms. So the design of this pilot was to ensure that we could obtain adequate tumor tissue and perform biomarker assessment. Patients with either melanoma and head and neck squamous cell carcinoma were identified. And from there, they underwent a biopsy and are assessed in the IMatch screening platform with whole exome sequencing, as well as gene expression profiling. We can say nanostring here because that is a long story, but that's what's being used. And there's a two-stage study. First part is that where people are undergoing both these analyses. However, treatment is being done prior to the results coming back. In the second stage, which we are not on as of yet, the treatment will be assigned once patients have the results of TMB as well as the nanostring TIS returns. They can be stratified into one of these groups. Primary objectives, I won't go all through all of these, but they mainly focus around feasibility. The ability to perform molecular characterization based on TMB and TIS with a less than 21-day turnaround, because this really would support being able to do a larger study. In addition, show an efficacy with the overall response rate in each disease cohort, as well as within tumor biomarker subgroups. And finally, secondary objectives specific to each disease, a cohort, molecular subgroup. This is a little bit complicated here, and people turn their head sideways for a moment, so I'll try and walk through this. So not only are we looking at head and neck cancer as well as melanoma, of which there's 60 patients in each cohort, but using the combination of TMB, but also TIS, we can break it down into distinct biological categories. This is somewhat pilot in nature and really looking for signal of activity and where these diseases might work best. Sorry, getting a page there. However, what's happening is based on these groups, we're looking for 15 patients in each of these groups, TMB high as well as TIS high, that is tumor infiltration score high, and then TMB low as well as TIS low. And you can see through the 4 categories what this falls to. Each of these represent a totally different type of immunologic tumor. We're using TMB as well as TIS as surrogates for different aspects of the immune microenvironment, although we could do more advanced characterization. We talked about how that would take much more tissue, much more turnaround. So these are representative in terms of segregating tumors. And we have the definition of biomarkers was identified before going into this and is identified there and is, as we'll talk about, a little bit of a work in progress as we go along. And it's been a great collaboration with CTEP and our partners in industry. In terms of the eligibility, advanced melanoma or head and neck cancer with measurable disease and being inclusive greater than age 18, ECOG, ZBROD 0 to 1. Progression after anti-PD-1 within 12 weeks of the last dose and naive to VEGF inhibitor. And then this third point we'll talk about several times in the next few slides, but adequate tumor specimen obtained after the development resistance anti-PD-1 therapy and within 6 months prior to registration. And this could be an archival tumor or a fresh biopsy performed for this trial. There's some restrictions around this, but this is a very, very important part. If a patient had an archival tumor specimen, for example, they had a biopsy after they progressed on PD-1 because there was concern about, is this truly metastatic site? Or they had a resection of oligometastases. These could be used and patient would not have to undergo a repeat biopsy. However, if a patient did not have a tumor tissue available since progression on PD-1, at that point, they have to undergo a fresh biopsy for the purpose of going on this trial. Also, no active infections, recovered from to grade 1 or less from previous treatments, and then washed out of CYP inhibitors. Due to toxicities from VEGF inhibitors, controlled blood pressure, which we are clarifying a little bit in a subsequent amendment, we'll talk about no prior radiotherapy to the head and neck region due to concern of fistulas, as have been seen in some phase 1 trials of VEGF plus PD-1. No bleeding, ability to take oral drugs. Drugs cannot be crushed, unfortunately. And then once again, a little bit busy slide, but keeping this clean. Patients are treated with a combination of nivolumab every 4 weeks and cabozantinib, and started at full dose, 40 milligrams per day. And there is a generous schedule to decrease based on toxicities. Based on that, as aforementioned, archival tumor fresh biopsy are required, and that's for treatment stratification in stage 1, which we're currently in. Treatment is started before these results are available. However, once we hit stage 2, the results of that those analyses will have to be returned before patients start therapy. Furthermore, there's whole blood obtained at several time points for translational studies. And finally, at progression, there's an optional fresh biopsy as well as whole blood that's mandatory. Study calendar is relatively straightforward and in brief, disease assessments every 8 weeks per standard of care, whole blood, and then blood pressure monitoring. In terms of statistical considerations, as it breaks it down a little bit here, for stage 1 and stage 2, we discussed looking at halfway enrollment to look at feasibility. We just have an interim analysis to make sure we were coming along well between stage 1 and stage 2, that our turnaround time was appropriate. However, we also have what's called feasibility monitoring every 20 patients to make sure that we are on track and that we're not getting extremely far behind in terms of turnaround time, QC failures, and everything else. And that's led to some really interesting findings that we'll discuss. There's also other formal interim analyses, including a TIS cut point analysis. It's a very interesting issue that we came into that a lot of times we don't think about with biomarkers. I think a mentor of mine and being at University of Michigan, Dan Hayes, brings up that a bad biomarker is as bad as a bad drug. And a lot of the published literature with nanostring we're using is either proprietary from Merck, at which point they can't actually give out their cut point. And not only that, it was in archival samples and first line, so it's not exactly the same. So we didn't go into this having an idea of the exact biomarker of what the TIS cutoff should be. And so nor did we know the exact average for head and neck cancer versus melanoma. And so therefore, we made this used the best data that was available, but had interim analyses to make sure we were mindful that if that was actually a median, and also to see if that really was predictive of which group. So I think this is a very novel strategy that was developed with CTEP to make sure that we were real-time monitoring our biomarkers. In terms of our study update, currently we're on a pause. We won't call it a hold, but we'll call it a pause as an amendment goes through. We have over 400 sites, as you can see, that applied. In terms of approved, 148. And while we're on this hold, we've had 27 patients accrued to the intervention phase. There's been several sites, especially that one on the first line that's put a lot of patients on, University of Michigan. But overall, we've had a lot of interest, and it's been very collaborative, a lot of discussion back and forth. It's been very nice to see involvement of both SWOG, Alliance, ECOG, and NRG, and a variety of collaborators. As aforementioned, we're on an interim hold. During this feasibility monitoring, every 20 patients, it was noted that optimization of procedures needed to be performed surrounding tissue acquisitions and processing. Match needed a smaller volume of tissue for their sequencing. Even the beginning of Match, they need a lot more tissue. However, when we're doing TMB as well as administering TIS, we began to realize that we needed a lot more RNA and DNA than past studies. We started to see that also biopsies were different facility to facility, also tumor type to tumor type, and worked very closely with Nationwide as well as with NCI, and noticed that we needed to standardize some of these issues. Also, there was been a delay in terms of validating a central lab for performing the NanoString assay. The CRO has taken some time with the federal government and an approval process, which I think would be a presentation onto itself. Once there's an amendment going through right now that's doing a couple things, including formalizing a change in the statistical plan that allows us to open up again, changes some eligibility for clarification, as well as helps with blood pressure monitoring and toxicity management, which have been a difficulty for some sites in our wording. And also with the when this reopens, there will be no reason to wait for TMB, TIS results. And so we're very much looking forward to this reopening. In terms of the processing update, we've been on average less than 21 days, which has been very reassuring for the entire team. However, you can see 84% have been less than 21 days. In terms of TMB high versus TMB low, initially these two diseases were chosen based on a retrospective paper suggesting relative equal TMB, TMB low between the two diseases. However, we've only seen 3 TMB high, all of which are melanoma, and the remainder of patients TMB low of 16, which is interesting. We're considering on an ongoing basis. I don't think we have the issue with QC, as I mentioned. Many patients were having issues with QC. We were having tumors arrive at Nationwide, and we're seeing either very little to no tumor, an entirely necrotic tumor, or very low DNA RNA. And we had a very good, thoughtful workflow here involving all the members from study coordinators to PIs to radiologists, and as well as partners from the original Match trial. And we saw that most of this, if we could isolate one issue, is due to insufficient tumor content after travel. Several factors likely could be leading to this. One is EBUS, when you're doing a bronchoscopy, getting a sample of lymph nodes. Sure, sometimes that can't be overcome, but you're getting a smaller sample there and often degraded as there's mechanical traumas you pulled out through a smaller gauge needle. Furthermore, when you're getting IR procedures with a smaller gauge needle, you're getting less tissue there. And end-of-week shipments, inappropriate shipping protocols have been an issue. It was quite educational, I think, for all of us when we saw that, once again, pick on University of Michigan so I don't make anybody else look bad. Our sequencing rate for head and neck cancer is 95% using the same samples. When we shipped them to Nationwide, we were seeing not great QC rates. We recognized one of the reasons is that just simply how patients were the media was being transported, how things were being dissociated. So we have started to educate sites a lot more on how important transport media is, how important just shipping the free sample, and that's helped significantly. So as stated here, education of lab staff, optimization of workflow has helped significantly. And since we've done that, we've seen a significant improvement in QC. We've also had processes where all 4 fresh cores that are taken for at the beginning of the trial are embedded in the same block for nucleic acid extraction, and that's once again helped significantly. And also lowering the minimum requirement for nucleic acid, and that's helped significantly without causing technical failures at the central lab side. We've also had more communication with sites, which has been very reassuring, both sides, all the way to radiology calling us with questions on where to biopsy. And then we have educational sides to go out to the interventional radiology teams, analogous to what Match did. And I think I have those on the next slide. And there's a biopsy manual, including a pre-biopsy scoring schema, similar to what was done in NCI Match. In terms of communicating biopsy requirements, this was developed for Match, and I think it was actually published thereafter, where it looked at the likelihood of yield based on radiographic appearances of the different lesions, and we've made this standardized now between in our patients as well, and helped sites. And I think just implementing these criteria have helped significantly. Also, avoiding biopsies when possible, necrotic or cystic tumor deposits, even if the largest tumor size, and directly engaging IR proceduralists regarding how important these samples are, the size, and the content, so they truly understand what's needed for molecular characterization. So some commonly asked questions that I flow through our email on a daily basis. The timeline for reopening. A little bit is subject to our federal government, because this has to do with an amendment that's going through. And so hopefully we don't get shut down again in November. But our hope is the amendment that's going through right now is cleared, and that, hopefully that will happen in the coming weeks. That's being resubmitted here in the hopefully in the next 2 to 3 weeks. And from there, if the amendment is reopened, if the amendment is approved, we'd be aiming for early December, but maybe as late as January. But once again, there's various human factors that could come into prolonging this. And we'll be sure to talk with sites as soon as we find anything out. We have a list of people who have been asking to be notified. We're excited to do so. Is the pending amendment blocking reopening? We have to wait for this amendment to go through. So unfortunately, yes. When we reopen, do we have to wait for molecular results to come back? Once again, we do not. And so once patients have biopsies, enrolled, registered, the patient will be able to start therapy. If the specimen fails, can patients still register to step 2? As of now, patients can register to step 2. That's the treatment part of it. But in the future, that's when the stage 2, we will have to wait for TMB and TIS, so the patients can get segregated into one of those groups before initiating therapy. Cabo cannot be crushed, unfortunately, and the email listed here is probably the best email to reach out if there's problems. But of course, both myself and Siwin have our emails on the protocol, cell phones, and feel free to reach out anytime. I think that's it. An extensive list of people here. I can't name them all, but thank you all so very much.
Is there any question for Paul?
I was just wondering, at any stage, are you guys planning to expand this to other tumor types, or is it going to just stay with head and neck and melanoma? For example, from a GU perspective, it's already standard of care in renal cell, but could it be considered for bladder, for example, urothelial carcinoma?
The pilot is just for these 2 disease sites to really show our ability to characterize, as well as, however, for the general iMatch platform, and it'll be discussed in just a little bit. There's proposals looking at across tumor types, and so I think that would be a great chance to evaluate the combination or other combinations there.
Okay. Thank you.
Yeah, right now the iMatch study team is not calling for concepts yet. We're not at this stage. Thank you, Paul. So our next speaker is Dr. Hossein Toby, and he is going to represent the Immune Resistance Committee from SWOG and talk about an initiative to consolidate the definition of resistant to immunotherapy. So just a little bit about Dr. Taube. So he is a melanoma specialist, and he got most of his training from University of Pittsburgh. and his oncology training, and initially as a junior faculty, and he was recruited to MD Anderson in 2017. And has been having a very successful career. He was instrumental in the development of effective treatment for melanoma patient with brain metastasis, established the standard how we treat these patients. He's also responsible, a principal investigator of the trial that led to the newly approved Opdualag, anti-LAG-3 plus anti-PD-1 therapy for first-line treatment of melanoma. So thank you, Dr. Taubey, for being here.
Thank you. That was a very generous introduction. I really appreciate it. Thank you, Katie and Simon, for the invitation. I'm part of SWOG. I'm usually on the melanoma committee, so I'm part of the family. I didn't expect the generous introduction, so I appreciate it. Although today I am here with my SITC hat, and I'm presenting on behalf of our committee, and I'll walk you a little bit through this. I did present, just for those of you who joined the monthly calls, this will be redundant. The goal is to just give you a very high-level overview of the effort, and the goal of today is actually to make this a bit of a working meeting, because we have specific questions we're bringing to this committee. Really excited to hear about the expertise that's kind of amassed in this committee so that we can really get some feedback on how do we get our efforts to move forward. And I think hearing from Dr. Kuderer and from Katie and Simon about the efforts already ongoing in the SWOG-IO committee, I think we have a really great grounds for collaboration here. Will I be able to advance my slides, or do I have to ask for, oh, this one, right? Okay. So yes, so I am, one of three co-chairs. So Harriet Kluger from Yale, Ryan Sullivan from Mass General, and myself have been co-leading this effort. And we have, we started, as you'll hear, and I'll give you a little bit of history, started with a workshop. And as soon as we put the workshop together, we realized that this needed a more sustained effort than one workshop. And that's why this turned into a committee at SITC. And the committee members are listed below, and they really represent kind of a wide range of both academia and pharma as well, which, as you will hear later today, are critical elements also in us being able to understand what resistance is about. And I have to say, the first resistance workshop was really centered around a simple question, and partly was driven both by the community itself and by the FDA, even with the fact that we have obviously thousands of clinical trials, and I'm not going to show you all these slides that we all see with how many hundreds of drugs are being developed and how many trials are happening. And as we know, a lot of them are kind of centered around reversing resistance and second-line therapy. And we're going to add drug X to PD-1 and try to prove that this works better than, PD-1 alone. And the reality is it's really hard to assess the impact of these combinations in the second-line setting without taking into account how those patients did on PD-1 in the first place, right? So just simply speaking, a patient who's had as a complete response to PD-1 and then ended up progressing biologically is entirely different than a patient that just blew through all immunotherapy-based regimens, right? So it's a very important aspect of this. And I have to say, again, the FDA was almost asking for help because people are submitting their results and saying, you should kind of allow us to go to a registration study with this 30-patient cohort that we show 40% response. Or what does that response really mean in the context of PD-1 resistance? And so we gathered together in April of 2019 at AACR in Atlanta, and brought together, as you see, a wide representation from academia, from industry, from other oncology groups and the FDA and the NCI, and really kind of started thinking, first, this is an effort across all cancers. So it's not about, all three of us co-chairs are melanoma people. Apologies, we should fix that. But we really care about, all cancers at this point. The other thing is that first workshop was about single agent. So we wanted to kind of bring this very complex problem into a very simple question to start with. And so the first, again, workshop was centered around single agent resistance and how do you define primary or secondary resistance. And we arrived at a consensus. You see the publication up here, it was in 2020. So the consensus definitions came up as follows. We felt that it was important to account for how much exposure patients had. So We called primary resistance patients that had at least 6 weeks of exposure and had absolutely no evidence of clinical benefit, right? So no, they either have progressive disease or stable disease for less than 6 months. We did recommend and require confirmatory scan, again, to kind of rule out any possible pseudoprogression. And again, we called that primary resistance. As you see, as opposed to secondary resistance, which is a situation where patients have exhibited some form of clinical benefit to prior PD-1. So either CR, PR. We considered stable disease for 6 months to be clinical benefit, although taking into account that it depends on the cancer specifically. And again, required confirmation with a scan at 4 weeks, again, to rule out any sort of pseudoprogression. We also took into account that, we have patients now being treated in the adjuvant setting. So if you're in the adjuvant setting and you recurred early while you're on treatment or less than 12 weeks after stopping, then that's more consistent with primary resistance. And if you recurred more than 12 weeks after, that was consistent with secondary resistance. And then we also accounted for the fact that some of these drugs, and especially PD-1, is coming into the neoadjuvant setting. So how do you place patients that have previously received therapy? And we felt that major pathologic response was already, again, exhibiting clinical benefit versus no response that was consistent with primary resistance. And then there was also one aspect that we had to account for, which is that patients could stop treatment for multiple reasons in the metastatic setting. So again, adjuvant or neoadjuvant, we know that it's going to be a limited duration of time, but in the metastatic setting, patients could stop for toxicity or for maximal benefit. So again, we kind of worked through those scenarios and how we would define resistance, primary or secondary. But the point here is, as soon as we published those in 2020, it was clear to us that those were consensus definitions first, and that's what we're going to discuss a lot today, but also that single agent PD-1 is becoming already retro, and already we're using combinations across the board. So we really needed to start thinking about how do we address the same question if patients have received combinations. And Harriet and Ryan and I kind of thought about this for a few weeks and kind of tried to come up with a format for the workshop, at which point we realized it's a really big bite to kind of chew on. So we decided that you can't say combinations are all the same. And we have situations where we have IO combinations. Those are the easy ones, kind of because it's all within the same kind of approach to activate that T cell. But there's IO chemo that's already approved in multiple malignancies. There are IO targeted therapy approved in multiple malignancies. So we actually did a workshop now that was separated into 3 groups, and we defined each of the resistance to each of those combinations. So you have IO targeted, IO chemo, and IO. The other thing we did is we asked for a lot of help, and the goal of showing you this is just to kind of reiterate and make it clear that this is a community-wide effort. We're trying to involve as many people as possible, and this is kind of a list of the people that were involved, say, in the IO-targeted therapy one that I co-led with Mike Atkins. Again, I'm not going to go through the list, but as you know, as you can see, it's again industry in addition to academia and NCI and FDA. Same thing for IO chemo, which was co-led by Ryan Sullivan and Nair Rizvi. And this was published as a separate manuscript. And then the IO, which was co-led by Harriet Kluger and Ila Charon. So once we finish with these definitions, I'm going to go through them. Again, it's not, you can refer to the manuscripts. I'm not going to quiz you at the end, I promise. But the idea is that, again, each of them will have a different characteristics, different things to think about. IO chemotherapy, if those patients have progressive disease in less than 6 months, those were considered, again, primary resistance. We took into account that this is a situation where confirmation with scans is not the same. If you're doing IO, we worry about pseudoprogression. But if you're doing IO chemo or IO targeted, the incidence of pseudoprogression is minimal, if at all. And so we didn't really want patients to waste 4 weeks to kind of confirm resistance. But for patients that are receiving IO, that's still a possibility. So a 4-week scan is appropriate. So again, showing you this just to kind of give you a flavor of some of the things we were trying to tackle. Similarly, for secondary resistance, again, the concept is still simple. If you've had clinical benefit and then you progressed, a different population than if you've never had clinical benefit. And again, similar exposure requirements and definitions of responses to each of those approaches. And then when we started thinking about the adjuvant setting, it was a lot simpler of a discussion because we didn't have approvals for IO chemo. So that was not a there was no reason to define that. We do have IO targeted in renal cell. Again, we do have some inklings of IO in melanoma as well. So this is kind of still an early field. And again, maybe to note here that for each of those definitions, we relied on what's FDA approved, what is out there that people are using currently in their clinics as the kind of the background of what we're trying to define. Similarly, we talked about neoadjuvant therapy, but again, the goal is not to go through each of those, and I'm happy to talk about them or to refer to the manuscripts, but it's a high-level review of what we've done. And if you've heard everything I've said so far, is that we thought about this, we discussed it, we arrived to a consensus. It was all consensus among people like all of you sitting in this room that have experience with these drugs. It was not data-driven. It's all by our clinical experiences, by our experiences with our clinical trials. And so I think it's a very good platform to start from, but we recognize that it's not data-driven. And that's why our goal of our discussion today is to kind of think about how do we now that we have the definitions, they could be great and they could be the basis of us kind of doing IO drug development moving forward in the field, or they could be just wrong, and we don't know that. So the idea is, can we actually collect our data that's already in existence and kind of validate our definitions, use those definitions to tell us if those data show that those patients that have a primary resistance, for instance, have a worse outcome versus patients that have secondary resistance, so that the future could be a data-driven approach. We may end up refining those definitions. We recognize that we may have to have maybe different definitions for different cancers, but until we get the data, we really don't know what the way forward is going to be. And the idea, again, was not to just have those papers out there. The idea was to kind of use that as kind of a functional model for IO drug development in this space. And so, again, the process is kind of summarized here. The phase one was kind of these definitions that were consensus-based, and phase two is today. That's why we're here. Phase two is to think about how are we going to bring these resistance data together, and we're here at SWOG because we thought a lot. We kind of thought about different ways to get this done. And the simplest way is, and I look at anybody from industry in the room, is that industry have in their coffers a ton of data. But if anyone has managed to get that data out of them, please raise your hand now, because that's really difficult, especially if we're going to tell them, we're going to bring your Pembro data and put it just right next to Nivo data and Atezo data, and we're just not going to care about the name of the drug. I think we can get there, and that is our hope, but so far it has been a difficult effort there. So that's why we turn to investigator groups like you, so that we can start the effort from places that are kind of purely based in academia as it stands. And then the hope is, more data will help us understand this better. So eventually, and I was just talking to Simon and Katie, that this effort that you guys have here could be really the nidus around which we kind of agree on the ways that we're going to do this, what are the data elements that we're going to collect, and then kind of be able to start that pilot and expand it, hopefully, to as many kind of data coffers as there could be. So this slide is the next steps, but honestly, the next steps are what we're doing tonight, to this afternoon, really. It's just kind of, how are we going to define the data elements? We've talked to ECOG Acronym Alliance. In all of our discussions, actually, this committee and SWOG actually have probably the most evolved effort to collect data from multiple clinical trials. And from our understanding with Dr. Kudur and her colleagues is that there's an effort around toxicity that's happening in that space, which is, again, that's a really great start, because adding the question of, response, outcomes of those patients, objective response, PFS, OS will be critical, so that we could really answer the question of, kind of are the definitions valid? If they are, that's great, and let's use them for drug development. If they're not, how can we refine them in a data-driven way? So, so this is my last slide. And again, we wanted to spend as much time today really talking through the data elements. So will we be able to pull up the Word document? Thank you. So, so this is the list of data elements that we came up with, and I will tell you why some are highlighted. But again, for any one of you who's been involved in data collection from multiple sites, the simplest guiding principle is simplicity, and being very clear about the minimal data elements that you need to answer a question. So we were trying to go with the, kind of the least amount of data that will be required, but will be able to give us the answer here. And again, this is the list that we came up with. I have a few questions for the committee. That's why these things are highlighted specifically. One of them is, for instance, date. As we discuss these, as you guys know, a date of therapy is actually considered PHI, and then there's a big question about, how much PHI can we use, or can we kind of share across different platforms? And then the second question was about the availability of RECIST measurements. But I'm going to really stop here. And again, this is up for questions, discussion, ideas of how can we evolve these data elements, anything we might have missed, and so on. So thank you for listening. And yeah, it's a working meeting. So
yeah, thank you, Hussein. So the SWOG leadership delegated this project to the IO committee, and we're just really excited to support SIGC and the immune resistant committee from SIGC for this project, because as Dr. Taubey mentioned, there are a lot of consensus out there about resistant, how to define it, what is primary, what is secondary, what is acquired resistance. But these are consensus, and there is no data to back up, and they can be wrong. And yet it's so important to have a consensus or a criteria that everybody agree. So in the future, we can actually consolidate the results and do more secondary analysis. So Hussein, I will let you to lead the question, the answer part. But I have a question for you. These are very important information you want to get from these completed IO trials, right? So what is the plan to go from these data you will collect to, confirmation or, to prove the concept that you are trying to promote?
Yeah, thank you so much, Simon. Again, I really appreciate the opportunity to be here to talk through this. I mean, our simplest question right now is we've actually, bucketed patients into primary and secondary based on how long they've been treated, what kind of response they got. So if you've ran a trial with 1,000 patients, with patients treated with single agent PD-1 versus, something else, and we have that data, and we can take each of those patients and say, okay, based on their clinical outcome, they've had 6 weeks of exposure, they've never had clinical response or benefit. We're going to bucket that patient into primary resistance. The next patient has had, a partial response that lasted, 8 months and then progressed. We're going to put that patient into secondary resistance. And then once we have those bucketed, then we're going to look at PFS, well, at OS primarily, because that would be the kind of the ideal final outcome, and whether that actually mattered or not for OS. And if it actually proves that, yes, the OS is very clearly bad for primary resistance, and it actually kind of, our definitions reflect reality, then that's validation to the definitions we have. If we find out that there's a lot of kind of gray areas between primary and secondary, and the differences are not as stark as we thought, we're going to then look at what are the elements that were predicting that difference, and then go back and perhaps refine our definitions for how we bucketed those patients. So that's kind of the, again, the overarching question there. Does that make sense?
Yeah, thank you, Hussein. Go ahead, Anne. Anne is the executive officer of our committee.
Really, really important to clinicians. So I really applaud this effort. We've talked, and to researchers. I mean, obviously, we always think about where do you have the cutoff? But I guess I see number 32, treatment beyond progression, and we do know there are lots of clinical trials that say, do you have clinical benefit? So how are you going to balance the clinical benefit versus resist progression?
I don't know. How do you think we should? I mean, we kind of felt that obviously that's a practice that's happened both in clinics and in a lot of trials. We're presenting at ESMO next week a trial for brain metastases where almost half of our patients received treatment beyond progression and ended up actually getting really a lot of mileage out of it. So I really think that's an important aspect. We didn't include it in our definitions because we're being kind of, again, trying to simplify how you bucket those patients. If it turns out to be very relevant, then we have to go back and kind of maybe refine exactly that definition based on did they receive treatment beyond progression and how did that affect their outcome. So, everyone, Takao.
Hi, Hossein. Thank you for taking this on. You know, the data is like scattered across different disease committees, right? And I think that's been a challenge for this committee for a long time, right? Because this committee itself doesn't have the actual studies. How are you planning to engage with these different committees? I saw on the slides that you mentioned, you know, you're going also to other cooperative groups. I mean, how would you do that in a collective study?
Well, I was going to rely on you for melanoma, Takao, but I mean, I think that's a great question. That's why we started our discussions with the chairs of ECOG Akron, the chair of SWOG, Chuck Blanke. Like we started there to ask them how do they recommend that we do it. And as you know, Siwan said, it was their recommendation to actually come to this committee. And I'm looking at Megan. We're going to rely on Megan to kind of extort the rest of the data out of the other committees. But you know, again, the point here is this is a community-wide effort. Again, SITC is putting in the kind of the structure for it, but I think we all need it for our, you know, again, future trials in the secondary and the second line space. And so hoping to get not just the consensus on the definitions, but also on the importance of this and that the different committees will see the value to their patients for that as well and for their studies. So it's a lot of consensus building and champions. And I think you were just volunteered for that, Takao.
So that's a great question, Taichao, and we're exploring it together. There are different ways to approach this. And, you know, we have Megan who will talk about secondary analysis just across different trials. And, you know, we're exploring the most collaborative way to get this accomplished. And I think this is a good example to show our members how these kind of concepts can be initiated and can be done. Hopefully initiate more concept coming using the same method, same process.
I have a question about Siwan's question earlier about your hypothesis going in, the goal of these buckets. I'm going to challenge a little bit saying, is it possible to say you're going to have buckets before you even start? Because I would argue each disease is completely different. And like one of the boxes up here that concerns me most is disease histology. And so the coding of that, just picking on one for a second, is going to be so hard to define acquired primary resistance, everything across diseases, much less with a single disease. And then your sample size per disease. And then not only that, you know, what is your actual endpoint there? How are we going to find things? So I'm wondering if you're making definitions even before you have enough data to say, does that exist? So that's my first question about has your group turned around? Is this going to be pan tumor? If so, why versus disease specific? And if so, what's going to be your cutoff? And then finally, does that really hold up over time? And I guess that gets to your statistician, but I would argue that if the decades over time, we know that, you know, survival has changed with disease biology. Can we really say that the disease biology of second line IO versus third line IO, these definitions, acquired resistance, primary resistance, are the same, or does it have to be different per type of therapy and per era? At which point, by the time you're making this, it's antiquated already. So, I mean, it's an amazing conceptual reference when you're writing a paper and we're thinking through trials, but what does this mean in the end of the day? And I think it's a great point. challenging what.
Yeah, no, I really appreciate it. And that's exactly what we've been kind of racking our brains around for the last, 5 years or so. Because the first time we met in Atlanta, the idea was, should we do disease specific or should we do kind of multi or trans disease, so to speak, kind of definitions. And so those were discussions that were had deeply among the committee members, and where we started was to kind of put everything together. If you read the manuscripts, which I recommend you do, there's a lot of footnotes, because we absolutely recognize that, for instance, diseases where the pace of the disease is slow is really, stable disease for 6 months. Is that really clinical benefit? Or the exact opposite, if you're treating anaplastic thyroid carcinoma, and those patients get 3 months of stable disease, that's clinical benefit, right? So definitely there needs to be adaptation. And so, our thought process right now is that, again, the data may actually be able to help us understand whether there's, specific diseases that could all follow a similar kind of definition. So, again, maybe lung and melanoma can be, and renal cell can be relatively similar, but maybe kind of other diseases will be different. And then, again, we're trying to engage investigators across the spectrum, so that if a group feels that, no, they need their own specific definitions, at least they can adapt what we have, again, hopefully with a data-driven approach to say, look, this is why we need to use these criteria. So, but I have to tell you, what was very clear to us back in 2018, as we were preparing for the 2019 workshop, is that this effort is not being done right now in a very kind of prospective, clear way, and we had to start somewhere. So, which is why today, like, I'm coming here to tell you, I actually don't know the answer. None of us does, but we're trying to start somewhere and to start to apply the most important yardstick, which is data, to tell us what is the right definition that we should use. And again, whether we should differentiate into different cancers. But thank you very much. We also, can I just kind of address one other point you made? Like, I am trying to stay away from, actual biology as much as I kind of can't wait to talk about biology, right? So the idea is, again, we've been asked this question, well, what about, do you want tumor tissue so that you can actually do translational studies to answer that? Absolutely, but again, if we kind of bucket people in the right clinical buckets, then we can ask translational questions that are a lot more pointed and can really help us understand maybe that there's commonalities in biology across the diseases.
Hussein, thank you very much. So we have a comment and then a question online from the people online. So first of all, Arnab Basu says, great presentation. I wonder if with respect to investigation, ctDNA is also going to be useful, as some resistance may emerge and then expand. So with respect to question, should we ask regarding availability of serum at different time points?
That's a great data element. We just added number 53. Thank you.
Great, thank you. Then we have a comment from Nicole Kuderer. Beautiful presentation, Dr. Twabi. An analysis will be valuable to start disease-specific based on well-characterized trial histologies, as well as avoiding buckets of resistance versus being initially descriptive about the outcomes data and what we see in terms of timing of response and progression. So that was a comment.
Agree.
All right, thank you. So next question.
I just wanted to make a little comment about the previous comment that was just made by the fellow from the University of Michigan. So this is reminding me very much, I was involved in a national effort for liquid biopsies, and we worked very closely with the FDA. And the first thing that we wanted to do was establish what are the most important things to collect so that the collection is done properly. Because if you don't do that first, it's not going to work. So we sat down with FDA and we came up with a list of 54 things.
We're missing one. Then.
Well, yeah, but you just told me of 53. So, but the point is that we all felt that it's too much. You have to be a minimalist here. And so what we did was we looked at things that were disease agnostic, tumor agnostic, and we made those And we pared it down to 11, and it became a publication, and it became quite successful. Now FDA gives that to companies when the companies come and sit down and say, what should I be collecting to prove that my assay is going to be in line? They said, well, the first thing you have to do is collect things properly and collect the blood properly, and then the things that we want with the blood. So it's just kind of reminding me of the same thing. Now this was very hard to do. It took about 3 years of a lot of meetings and a lot of grinding of teeth, but it did get done, and it was quite successful. So
May I enroll you as a consultant then? Because we're gonna definitely need help. And again, Megan's gonna speak right now
Sure,
about the.
I'd be happy to help.
That'll be great, really, because exactly to your point. Yes, we have 50 elements, but when we were looking through them, it's like, what's the minimal number that you need to actually answer the question? So, what people were treated with and what kind of response they got was, critical to answer, are they primary or secondary? But this is the idea. And again, the goal is not publications. We published the stuff already. I think it's really the usability, the actual ability to help with drug development and to change the way that we do it. And I have to tell you, again, in melanoma, again, that's the field that I'm most familiar with. There's been already people publishing their data by using those definitions and showing some differences in the response rates based on whether primary or secondary. So, and I've seen protocols already come through with these definitions even from the first paper. So we're hoping that this becomes a, kind of a handbook of sorts into how you kind of develop your next trial, as you said, like hand it out to industry and say, this is the one you need to. Great, thank you.
I just want to add, given the complexity and the number of elements, you may want to, at least within SWOG, consider a pilot for one tumor type and see what barriers you face rather than branch out and start with everything and then try to address those barriers and then maybe spread it out to other. Because even within GU cancers, like the definition of resistance to the point that was raised previously, within RCC and urothelial, the definition of resistance is very different. So you, just as a suggestion, you may want to pilot it in one tumor type, see what barriers you face, and then maybe expand it to the rest of the tumors, because I think it's going to be very hard otherwise.
Thank you so much. We'll definitely take that into account. It's always a kind of a balance between, do you focus on one disease, and we don't want people to think the goal is one disease, right? And so on the other hand, to your point, feasibility is very important. So yeah, I mean, we're hoping, again, when I presented the first time, it was great to see from Nicole and from Megan that there's been some efforts to collect data that may actually help us, again, make that decision of whether to move forward with specific diseases or just continue to be aspirational, so to speak. Thank you. Well, this has been very helpful. Again, I'm waiting for element number 54 and any other elements you can think about. And again, thank you for the opportunity. I really appreciate being here.
Yeah, we're really excited about this project, and we look forward to continue to contribute. This nice discussion lead to our third talk by Dr. Megan Otheus. She is a professor of biostats from Fred Hutch and University of Washington. And Megan is the smartest woman I've ever known. She got her PhD of biostats from Harvard University, and she has been the lead statistician from the Melanoma Committee, the Rare Disease Committee, Leukemia Committee at SWOG, and she has helped many phase 3 trials that led to practice changing results, and she's really an expert on secondary analysis. So today, so at the last spring meeting, Dr. Michael LeBlanc actually talk about, databases as SWOG that we can utilize for secondary analysis. So Megan's today is going to talk about how to utilize those results to do secondary analysis. So thank you, Megan, for being here today.
So yeah, well, thank you for inviting me. Secondary analyses are like one of my favorite things I get to do at SWOG, because with the primary, you put all that effort in, to run a trial, and then you write the one paper, and then that's it. But you can do lots and lots of other stuff with the data. So I'm excited to talk about that. And I wanted to, the second half of the talk, I think we'll get into like some more of the very specifics. If you already know that data is there and you want to access it, what those processes are. But for anybody who has questions that are like a little bit bigger, I'm trying to see, how can I, with the pointer. Okay, there we go. Okay, so the first thing that you need is a specific research question, which seems really basic, but I spend a lot of my days helping people realize that the research question isn't specific enough. So this is a research question that I might get every couple weeks from somebody who's like, SWOG has this data, let's look at it, but it's not specific enough. It just doesn't, SWOG does have data, but SWOG has a lot of data. So like, which is the data, and where can we, where do we have data where we can really make conclusions? So as an example of a specific research question, this is actually something that I've written up and I'm going to submit in a couple weeks, where I wanted to look at like kind of the same thing, but I narrowed it down to one study in particular, and I narrowed it down to a specific time, just the first cycle. And also, in terms of like adverse events, I took all treatment-related adverse events. But so anyway, it could be any of these things, but you can see like all those elements. We kind of need to know all of those when we go into trying to help you get access to the data and do these analyses. Okay, so one place to start, if you're like, does SWOG even have the data? So this file, the file here is kind of big. So if you do the QR code, it takes a while. It's just because it's big. So SWOG, so we at the Stat Center put together this report of studies, which you may or may not see because it's just online. It used to be like a printed book that you could pick up here. And now it's just online, and it has summaries of all the studies divided by committees that like run trials. So it kind of shows you the span of them, and each one here, it's really small. There's a table of contents. This is for leukemia. And then every study has a schema and gives you information about it. So if you're newish to SWOG, or you spend time with SWOG, but kind of in one committee, and you want to get kind of a perspective of the portfolios and other committees of overlapping, this is a place. The summaries are kind of written in a high level, or just like a couple pages. And in general, they show like accrual and toxicity for each study. If you want to know more about a study, though, you might want to see the protocol. Those are all available on the SWOG website. If you registered for the SWOG meeting, you should be able to access them here. Okay, so SWOG has trials. Now what? So there's kind of two paths here. One of them is who's going to analyze the data. So you want to work with somebody at the SWOG Stat Center, or do you want to get the data set and work with somebody else? And those paths have kind of different approval processes and mechanisms. So if you don't want to work with SWOG statisticians, if you've got another group that you're working with, something, or you're a statistician yourself or whatever, if you want to get it, well, we're funded by the NCI, like we share data, like that's like our, we got to, and we want to. So there is a template. It's about the proposals they can be long, but they don't need to be. They can just be, usually there's a template where you fill in a couple paragraphs about background, the data you're requesting, like the specific studies and the data elements. The proposal has to be pretty specific though. This isn't a proposal where you come in saying, I want to look at toxicity at all IO studies. That's not specific enough yet. Like you really, like this is that template they review, and most of it is a cross-check. Do we have that data, and is any of it kind of embargoed and in the process of being published? And that template is what is used to generate the data use agreement, the legal agreement for us to be allowed to export the data. So that's why it has to be really specific. It goes through executive review. And then after that approval comes in, the SWOG legal group, they are going to start with the data use agreement generated on the SWOG end, not on another group's end. They're going to contact, and then they'll follow up with whoever submitted the proposal to make sure they have kind of all the legal documentation, that you have the protections in place, you're allowed to analyze the data oftentimes, and especially if we're talking about a data sharing request that has multiple studies, the NCI is going to review, and there could also be companies that review it depending on like the terms of the CRADA or however they supported the trial. And then when all the signatures are in, SWOG sends you the data. We send it in pretty much any format you want. In general, from my end at the Stat Center, when these come in on my committees, as soon as we have executive approval, we start working on putting those data sets together with data dictionaries. We contact you, figure out what format you want them in. And so when the signatures are in, oftentimes we can send you the data like the next day. So it's not like we don't start working on it till everything. We try and like realize that sometimes that kind of living in that the legal land of getting all the signatures like can take a while, but on our end, we try and be timely. If they're really big, they can take a lot of time. It does take quite a bit of time to put the data together. And data use, if you request data, you can request big, even if you know some of the data are embargoed, and they will put that in the data use agreement. Essentially, like we'll give you these studies now because those are available now. And when the publications for these other studies come out, we send them to you, and then we just kind of send the data as it evolves. So it's pretty flexible. It's not a very long template. It tells you where to put everything. It's not very hard. But the big thing is that you need to know specific studies, and you need to know specific data elements that you want. This isn't a place where they're like figuring that out for you. Okay, so if you want to work with SWOG statisticians, again, there's kind of your different paths if we're talking about one committee or more than one committee. So if it's one committee, that's like clearly straightforward. You talk to the committee statistician and the committee leadership. And different committees have different processes. So we're funded by the NCI, and we have, as statisticians, we don't just do data sharing and secondary analyses. In fact, secondary analyses are oftentimes a lower priority versus like opening a study and like monitoring it, publishing, you know, the definitive results of the trial. And so we have like a portfolio of things that we're working on. This is secondary analyses are a component of it. Different committees have different ways in which they prioritize it, which has to do with kind of the bandwidth and also just the amount and volume. So for example, in the leukemia committee, somebody just kind of like will email me a question and I'll say, yeah, we got data, or we don't. And we loop in leadership and let them know. And it kind of just goes in the queue. And when we have time, we work on it. There are other committees that have really formal processes for this, where essentially they say, we're going to work on this many secondary analyses a year. This is our bandwidth. And there's like an application process to do that. So but you can't know that. You can't know that until you kind of find out from committee to committee. So if it's one committee, you just kind of work with them, starting with either the committee statistician or the committee leadership, and they'll tell you what the process is and how to figure it out. If it's more than two committees, I say start with one committee, not to necessarily do the work, but in general, for these sorts of projects, you're going to have like one statistician or like a group of statisticians, a team who are going to be the primary statisticians working on it. So you're going to figure out who that is, and they will help get the data all together from the other committees. So while I sit on several committees in SWOG, I have access to all of the SWOG database. So that's not an issue. But who's the right committee to start with? Oftentimes with me, it's the committee that has the most data or the committee that has the most energy for it. If you don't really know, I think a really good place to start can be the SWOG exec officers who sit across multiple committees, and so just kind of have a sense of the different cultures and energies and other things, and like where the best home for your kind of multi committee like project would be. So if you have a really large idea that takes like a lot of time, maybe you want like essentially like you want to know that you have some statistical time that's like really like a fixed amount and a significant amount of effort. There's two, so we need some more funding to do that, right? Because if you're just taking it off of our usual NCI funds, you're going to be competing with the conduct of trials and the needs there. One of those is that Hope Foundation has provided a bucket of money that is, there's like an ongoing RFA. There's no like deadline. So when you have an idea, you can submit it. It's like a pretty casual application. The QR code is for that. You can also write a grant, an R25 or an R01 are the common ones, and include the statistical support in there. I've been part of a number of grants that have been incredibly productive because when you put the effort together to make one big data set for your one question, you now have this like great, well-curated data set that really oftentimes there can be a whole sequence of research questions you can look at and think about. And so a grant is a very tidy way to kind of get that, like looks good for you, but it protects your time, it protects the statistician's time, and kind of gives you the bandwidth. And an R25 is like 6 pages. It doesn't take that, or an R21, it's like 6 pages. It doesn't take that long. And for a couple years, I've been on some, we churn out 10 or 20 papers and those, you get your data set and then like you can just kind of keep running through it. Another question people will have, well, does the trial have, and there's usually a question about a specific type of data, especially molecular data and PRO data. In general, if we have those data, they're probably going to be an objective in the trial. And so clinicaltrials.gov will tell you. You can also just check the protocol. Again, it'll be in an objective. Sometimes some of our objectives, because there's like some like clinicaltrials.gov negotiations, sometimes some of those end up in appendices, some of the translational medicine details. If you have questions about specific samples, that's always in section 15 of our trials. If you saw if you think we have molecular data because you saw it in a publication, you're right, we do have it because it's in a publication. We will either have the data or know who has the data and know how to get access to that data. If there's molecular data and it's not published yet, usually the people who generated the data in SWOG have kind of like dibs on that first publication with the data. But once that data has been published, other people can request and use it. And the statistician who was involved with it should kind of understand and know the strategy of that. And then getting back at some of like the questions, I came in partway through Hussein's presentation before. Many of the elements, like one of the great things about working with SWOG, I think, and one of the things that really aids in secondary analyses is that many of the data elements that we collect on all studies, we collect the same way on all studies. So we collect survival. Survival for every SWOG patient lives on like one variable, like it is there, it's all together. We collect toxicity by cycle in standard ways that are the same across all of our studies. The way, if you have a trial with a RESIST endpoint, we collect RESIST data in the same way across our RESIST studies. So that data element and like putting it together, it still like takes time. There's still like it seems like it should all, but it does take time to curate it and make sure things match up. But I think that like really where the challenge is just is really framing the question specifically enough to make sure that you're getting the trials that are appropriate for your question in SWOG. So reasons and analysis isn't feasible. So while I love doing them, lots of times people email me and sometimes just like, we didn't collect that data. And there'll be like little flurries. You can tell there must have been like, a presentation at a scientific meeting I wasn't at, because I'll get a flurry over like the next 3 weeks of questions from people all over being like, did you collect this data on this trial? I was like, nope, sorry. Too bad, next time. The other thing can be that sometimes our sample size is too small. This can happen with, for example, there have been a number of requests about really specific rare toxicities in like with immunotherapies. Thinking like, well, SWOG has lots of data, like we'll have, but it's true, but still sometimes those rare events are so rare, we still only have like 4. So like, that's just still at a descriptive. There's no statistics to do with 4 we just described. Related to that is not enough events. When we're talking about time to event endpoints like overall survival and progression-free survival, our precision comes from the bad events on patients, not the sample size. And so just being aware of that, we might have 2,000 patients, but if you only have 10 deaths, you still don't have a lot of precision. And then the biggest thing is that people come with research questions that aren't specific or clear. And then we just like spend years circling, and actually I'll like analyze, and then it won't be right, but we won't know why, you know, like, and it just like is a circling endless loop of not very productive for like anyone involved. So I'm happy to take questions or emails about any of this. If people have specific questions, right?
Wow, this is so helpful, Megan. So I learned a lot from your talk. I think one of the things you're trying to convey is get a grant funded, then it's easier to get
If
the
you
data.
have, yes. I mean, and there's Hope Foundation money, but depending on some of your scales of projects, like if you really have a vision that could lead to like a lot of papers, I think a grant is like the best way to get like a dedicated set time with like the people who can get the expertise on it. Like the kind of approaching committees, I think that's most effective when you have like a one-off little project where it's like, can you just look at the association of this with outcome? And we do the analysis, like, you know, it takes me a half hour, we get tables and plots, and then like we spend some time to write the paper, but like it's just kind of a well-contained. But when you're talking about the bigger ones that are kind of making big cohorts and putting that effort in, usually you have like a sequence potentially or a number of ways you want to interrogate that data. And in my experience, those have been, they've been more easy, they've been like more generously reviewed by study sections than translational, than like my translational medicine proposals. And on average, we have the data, you know, like it's much more concrete what we go in and what we can say we can do, because we can say we have the data. There's not any ambiguity. It's just like the time to put it together and publish it.
Right. So if several committees are involved, even for published data, you still need their like a formal approval, like written
Yes,
approval.
so SWOG has like rules. So essentially the committee chair gives the approval in general, and then depending on the publication, I'm just submitting this week in leukemia, a big one, and we have like data from all over the world to look, because we wanted to look at this rare endpoint, and you just needed lots of people to be able to have enough data to talk about it with precision. And the study chair of every study involved is also, they usually aren't involved. They're like just at the publication, they get to kind of be added on and reviewed, but like SWOG in general, if you're a study chair, you get to still have you get to receive credit in that way. And then the committee chair is the one who really is like giving the sign off. I don't know for these IO things, if like Chuck kind of supersedes the committee chair. So when Chuck says yes, then that just means we have to figure it out. So I think then the question is figuring out who would be the lead. Like it would make sense to have one person be the lead, but also figuring out which of our studies, and which parts of our studies, like, are you going to take phase 1 studies, or do you just want further phase studies, and, you know, all those things.
Yeah. And one last question from me. So Hussein has 53 different things he wants to collect. What if it's not collected by the trial?
Then we just say we don't have that.
There's
And then we
no-
just give you everything we do is typically what we do. People give us, like in those data requests, they give us their wish list, and then before it goes to triage review, usually they flip it back to us and we just check off the ones that we have or don't. And they will essentially say, we have these, do you still want to go through the hassle of like all the legal stuff to get the data, knowing we didn't collect X, Y, and Z? And typically people say, yeah. So in general, like ask for it all, and then you'll just get what we have. Nobody's gonna, yeah.
So there's not the possibility to go back to the sites, even if they want to collaborate.
Then in general, in the past, that has required us paying the site for their time to give us the data, which then usually requires some sort of
Funding.
grant or other funding, pharma funding, like somebody to fund the effort at the sites for that request. Sometimes that information can be, depending on, like in some of my studies, it's not that we need to go back to the sites, but it's that we didn't ask for it on a form, but we have like source documentation that may have that information. And then in general, people just like volunteer their time, like like you get your little team of doctors together to like curate and like kind of create some elements. We have done that before. I think here it would be, if you really need data from a site, we would just need to probably provide them funding to,
Right.
So a quick logistic question, and thank you so much, Megan. You always remind me how much work this is going to be. Thank you. But just in terms of, so you have the data, I'm assuming it lives in CRAB or whatever it is, but then have you ever collated it with data, say from ECOG Akron or from external sources? Like if you had to put data together beyond what you have, how do you do that?
I do that more often than I would like. Like this project I was talking to you about, I have data from the leukemia group in Australia, in the UK, in Germany, in Italy, Alliance, ECOG declined to participate, Alliance. And then we have data from a couple of large single institution cohorts. So anyway, that also just takes time. And so the fewer, like if there are fewer elements involved, then you kind of can tell people, give it to us like this, and then it can merge together. I oftentimes don't want there to be like a barrier to people giving us data. So I'll just say, give me the data, and then I like figure it out. But essentially then you just have to map it to make sure things line up, and it takes a little bit of extra time. But yes, it's very doable. We do it, regularly, or I do it regularly. Other people are like, I don't want to do that. That is a lot of work. And I was like, I know, it's like kind of stupid, but no, but I think that is, it's totally doable. Yeah.
Great, thanks.
Thank you very much.
Thank you.
Thank you. So we're going to move on to our final presentation of today's IO committee meeting. And it is my pleasure to introduce Dr. Marleen Kok from the Netherlands Cancer Institute. Dr. Kok is a group leader at NKI, where her laboratory and her clinical efforts focus on translational breast cancer immunology, including work that she does to lead innovative immunotherapy trials in the context of breast cancer. Dr. Kok has been a group leader at NKI since 2019. She did her PhD at the Netherlands Cancer Institute, working on breast cancer biomarkers, and did additional training following that at NKI and also Johns Hopkins, really focusing on cancer immunology, T-cell biology. And so it really is a pleasure to have you here this evening for you, I know, in the Netherlands, and this afternoon here for us in Chicago. So thank you very much for joining us today.
Thank you so much for a kind introduction and wonderful invitation. Yes, indeed, it's evening. It's almost 10, so I did a full day of clinic, so I might not be at my best, but still hopefully energetic enough to share, concepts and data, and not because I can envision that, at SWOG, the trial sizes are usually pretty large, but this is like the step before a large trial, I would say. So let's see whether I can control the slides. I'm not sure whether I am allowed to. I see a slide. Okay, something is happening. Does this work?
Okay.
Yeah, better. There we go. Okay, so the talk will be focused on breast cancer. So I do 50% of my time, I see breast cancer patients in the clinic. So my research is really breast focused. Though at NKI, which is one thing, a good thing that I really like about the place, is that as you know, immunotherapy and immunology investigators, we are really grouped together. So I work in the breast team, but we also have this immunotherapy team. And I share my office, for example, with Christian Blank, who runs the neoadjuvant immunotherapy program in melanoma. So that really, you know, creates an early crosstalk, but if we have the data, clinical as well as translational. Okay, but for those of you who are not, you know, fully on top of breast cancer, so we of course have the 3 different subtypes based on 3 measures. And I really think that this is kind of a stupid definition, measuring 3 things and then pulling all patients together while knowing that there are really subtypes of triple negative breast cancer. So I state here, no targeted treatments available, though we have PARP inhibitors, of course, but I think especially in metastatic disease, yes, the response rate is pretty high, but the responses are usually not durable. And of course, now we can also treat with anti-PD-1. Women are frequently young, and it's still, as of today, the survival, despite all new developments, is very poor of most women. So, of course, we are here talking about immunotherapy, and I think if you do the most simple assessment of immune infiltration TILs, you see the difference by subtype. With the triple negative, where we have the highest clinical need, having you have more chance that you have a high TIL tumor. But also you see that for HER2 positive and even ER positive, they're highly infiltrated tumors. And that brings me again, you know, to one of the most recent publications. It was done by an MD PhD student, and it was her idea, and it's wonderful if such a trainee can bring a project from idea to a very interesting publication. So what we did, giving the subtypes and knowing that, especially in the ER positive subtype, we've seen huge spread in amount of lymphocytes. We wondered whether does it matter what the level of ER expression is. And for those of you who work on breast cancer, we know that, you know, we have triple negative on one hand and ER positive, but it's 50 shades of gray depending on the expression of the estrogen receptor. And if this 50 shades of gray, not only on biology side, but I think also on clinical outcomes of patients. And you can see here that, you know, triple negative, ER low, and here are the different, you know, selected subgroups based on ER. And it seems that, yes, you are ER positive, ER expression between 10 and 50, but TIL levels seems to be as high or as low as in triple negative. So maybe this classical definition of ER positive, HER2 positive, triple negative, in the era where immunotherapy developments are going very fast, it's a very old-fashioned definition. And on the right, you see pretty much the same story if you do an assessment for PD-L1. So if you are ER low or ER intermediate, you're way more likely to be PD-L1 positive. And just a recap before we take off on the data that we've been generating in the last years. On the recap, where does IO stand in breast cancer? So for ER positive, which is usually, you know, although we appreciate this spread in immune infiltration, it's the coldest tumor type. The data are in phase 3, and next week at ESMO we will see data from phase 3 trials in a neoadjuvant setting for ER positive breast cancer. And there are large phase 3 trials ongoing also for metastatic ER positive breast cancer, usually selecting patients, for example, based on PD-L1. So to me, the developments in HER2 positive breast cancer, it's a bit puzzling. So there are immune infiltrations seen. The mutational rate seems to be similar to triple negative, but so far the results of anti-PD-1 based immunotherapies are rather disappointing. And I think we still don't know whether that has something to do with HER2, whether it's HER2 driven, or whether those antibodies like trastuzumab and pertuzumab are doing something bad to the tumor microenvironment. I think it's an important question that should be investigated. And then last but not least, where we have the highest clinical need, so in triple negative breast cancer, you see that it's standard of care together with the chemotherapy. But where are we talking about? Standard of care in a neoadjuvant setting. So these are the data on pathological complete response in the Keynote 522 study, stage 2 and stage 3 triple negative breast cancer. You see in the early analysis that the PCR increases with 14%, which is modest, but we know from triple negative breast cancer that PCR translate pretty well to event-free survival and overall survival. So the survival benefits so far seen, it's about 8%. Maybe we expected more seeing those PCR data, but I think the data are still not super mature yet. Though I think you have to realize that, we add anti-PD-1 on top of chemotherapy. And, in my perception, the chemotherapy neoadjuvant for triple-negative breast cancers, it's pretty tough. So we administer 4 types of chemo, so anthracycline, cyclophosphamide, carboplatin, and paclitaxel. It's about 5 months of treatment. And usually patients, they stay on their feet and rarely we have to admit them to the hospital, but still it's a huge impact on their quality of life for at least those 5 months, and it takes months to recover. So it's really the question, what was the next step in those treatments? So what is the optimal chemotherapy backbone? So I think we really take the approach, okay, the standard of care, and we add anti-PD-1, but is that the best approach? And that's what we have, of course. But which chemotherapy is key and will be the best dancing partner to go along with anti-PD-1? And as said, so it's still that 30-40% of the patients don't reach the PCR with this heavy combination treatment. So you need to study novel, ideally immune modulatory approaches to make sure that those patients can reach a durable response. And there's a key question on who benefits. In landmark trials, so the Keynote 522, we've seen data on PD-L1, which is not a predictive marker in that setting. But the biomarkers, mainly for pharma trials, I think at least the data are very slow if we see any of them. And then, we consider them exploratory, and I think it's very difficult to, if you have the biomarker data to make sure that it can change the guidelines for all kinds of good and bad reasons. So 3 open questions, and what I will do in my presentation is show you examples on how at NKI we try to approach it on a conceptual basis. So I will show you data of a proof of concept trial, which has the nickname TONIC, which is really focused on this chemotherapy and priming the tumor microenvironment issue. And second, I will show you how we try to integrate at our institution the mouse model data with the clinical data. And last but not least, some neoadjuvant data, which is of course known where the immunotherapy is most effective, and how we try to enrich for a biomarker population, which has the nickname the Bellini platform trial. So for those of you who have been working on this topic, so chemotherapy and what it is doing to the immune system, I think the literature is very complicated. So I finished, I was doing my fellowship back in 2014, and working with the guys in melanoma, seeing those beautiful responses on anti-CTLA-4. So as a breast biomarker interest, I was like, okay, what will it do to our patients? And we give the chemotherapy, and what will be a good positioning of the anti-PD-1, with no data available on checkpoint blockade in breast cancer so far. And studying the literature, I think the literature is heavily biased towards positive data, people using their favorite mouse model with their favorite chemotherapy, presenting their most favorable data. And that's why the data is, I would say, unorganized all over the place and difficult to compare, since for chemotherapies, different regimens are used in different mouse models. But this is, I think, an elegant review paper by Nacho Malero from Pamplona. And it sums all the pros and cons of chemotherapy and what it could do to different types of cell death and different types of immune cells. But it's highly simplistic, and I think it's rather complicated. So with this kind of pessimistic view on this topic, I would say, we were like, okay, we are not going to, if we try not to make the literature more messy in the preclinical setting, but let's see whether we can study this in a systematic manner in a clinical trial. So this is really a proof of concept trial, where it's a small basket trial, where the baskets are not based on a certain biomarker, but the baskets are different because we use different priming treatments. So we started in 2015, and basically it was a phase 1 concept. So all patients that enter had chemotherapy refractory disease. And the only option they had was basically like phase 1 programs. So it's not that we are studying here tumor control by an induction treatment. It's really low dose radiation or a certain chemotherapy that has with the aim to see whether we can change the tumor microenvironment. So we treated with an induction for 2 weeks without anti-PD-1. We took a second biopsy, and then all patients were exposed to anti-PD-1. And here you see the result if you pool all baskets. So this was the first set, so stage 1 data with 66 patients with an overall response rate of 20%, which is for this population relatively high. I have to say that we did select over the years, we tweaked a bit the inclusion criteria to avoid those patients that won't have any benefit. For example, we excluded patients with super high serum LDH, of which is known that the prognosis is so poor. I think that's why we are relatively high in response rate. But then if you split the response rate by type of induction treatment, you see differences. And of course, the numbers of this data set are still very small. We are currently expanding certain baskets, but you see at least differences that if you pre-treat with low-dose doxorubicin or low-dose cisplatin, it seems to be more favorable compared to the radiation. Of course, it's only one radiation schedule that we used, or low-dose cyclophosphamide. And next step, what we did besides, enlarging the series, the concept is that the clinical data should be supported by the translational data and vice versa. So when they go hand in hand, and we hope to get a better understanding. So that's why it's key to look on the same side, to look at the clinical data and at the same time at the translational data. Here you see gene expression data. It's still bulk RNA sequencing. The single cell data are now coming in. And what we did is we made the signatures of which we know that they are favorable, not only in breast cancer, but also in other tumor types. And if you see a red square, it means that after induction, it's an upregulation of that gene signature. And what you can appreciate from this graph is that the red square, so more upregulation of favorable gene signatures, it's more often seen after the chemotherapy induction compared to the control or the radiation arm. If you look at a bit further time points, so the patients are on anti-PD-1, they received 3 cycles, so after 6 weeks, we did another biopsy. You see that this difference are even more pronounced. So it's 6 weeks after the priming treatment. You see that here, you see that the difference are getting more pronounced, and the same as we see here, so less change in the control group as expected, as well as in the radiation arm. And next step, we performed TCR sequencing, again on bulk, and we approached this by making quadrants. So what you would do with your priming treatment is you want to have more T cells in, so getting, an arrow going up, and you also would like to have a more clonal repertoire, suggesting that something is getting recognized by the T cells. And if you then look at the difference between like the control, radiation, cyclophosphamide, cisplatin, and doxorubicin, of course, you see it's difficult to directly compare, but at least you see that here for those, the data is all over the place. While with cisplatin, most of the errors go up, as well as with doxorubicin, and you also tend to see more errors to the right. So as said, so can we validate those findings in more patients? And we just finished the recruitment of this validation set, so hope to present the data next year. And we also now, since this bulk data is also giving a glimpse, we are focusing more on complex tissue imaging together with a group of Mike Angelo at Stanford and with Christina Curtis on board. Single cell sequencing, the data are coming in. So we hope to get a better understanding of how those, chemotherapies could be good dancing partners for the anti-PD-1 treatment. And even we try to follow in the blood certain kinetics of neoantigen-specific T cells, which is, of course, very labor intense, but I think in an academic trial, we are in a position to do so. Okay, then to the second approach that we are taking in an academic setting and, getting the most out of an academic clinical trial is that, we combine the analysis with mechanistic studies in representative breast cancer mouse models. Here you see the team. So my partner in crime is Professor Karen de Visser. So Karen de Visser is an immunologist and mouse geneticist by training. She did her postdoc in the lab of Lisa Cousins, and since more than 15 years, she really believes that myeloid cells are key in the anti-cancer immune response. And she studies inflammation already for years. And she's been one of those few that 15 years ago, everybody would say, like, what are you doing? Why are the immune cells important? And I think now later, she was right. And it's nice to collaborate with her. And the team really consists of people working in her team, as well as people working on the clinical side. So how we do approach this, it's basically a co-clinical trial. So here you see this metastatic triple-negative breast cancer patients that I showed you before with combinations of chemo and anti-PD-1. We did set up a pipeline for fresh flow cytometry of the blood. It's very complicated to do so, but we strongly believe that if you're interested in myeloid cells and you rely on frozen samples, that you miss your favorite immune cells. So by freezing, you lose your neutrophils and you also lose vitality in your monocyte population. So all flow cytometry was done fresh. This is, of course, a single center initiative, making a bit more easy to do than you do this in a multi-center fashion. And what we did, we mimicked the same in the mouse modeling, the same composition of the flow cytometry. And then, of course, if you have certain observations in your patients, you can do intervention studies in the mice model and validate again in your patients. So that was the setup that we started years ago. And I said the main interest when we started this initiative was on the circulating immune cells, and especially the myeloid part. And this was one of the first results that we generated. And we recently published this story in Cancer Cell. It's kind of disappointed. So we looked at baseline on what kind of immune cell population correlate with response in metastatic triple-negative breast cancer. And you see that there is no correlation of any of those, immune cell subtypes with response. However, since we, performed this analysis in a longitudinal manner, the next question was, of course, do dynamics matter? So are there certain immune cell populations going up and down on response? And here you see, the intriguing result that in responding patients to anti-PD-1, the Tregs that circulate go up, dendritic cells as well as eosinophils. For dendritic cells, I think there's literature that's indeed it could be the case. For Tregs, the literature is a bit contradictory. And we were mainly intrigued by these eosinophils because it was really a surprise to us and unexpected. And why it was so important to us, because in this co-clinical trial, we basically saw the same in the mouse model. So the mouse model is a p53 ECAT here in the mouse model that makes triple-negative breast cancer that are particularly cold. And if you treat those mouse models, those mice with immune checkpoint blockade, there's hardly any response. But then if you add platinum-based chemotherapy, so in blue, you see that the survival of the mice gets better. So this is considered a mouse model good for chemoimmunotherapy. And if you block the CD8 in this setting, you see that this durability of response is affected, so suggesting that indeed we are having a T-cell based response here. And in the mouse model, so looking at the flow cytometry, you see the same as what we saw in patients, that mainly eosinophils go up if you treat with chemo and immunotherapy. And that's why I embarked on studying eosinophils. So there are bits and pieces in the literature on the role of eosinophils during cancer and during immune checkpoint blockade. Even in old days, I've been told that physicians that treated melanoma patients with IL-2, that they saw an increase of eosinophils. It was an observation. They didn't have any clues why that would be the case. But there's literature supporting that an eosinophil can be pro-tumorigenic, even shape a tumor microenvironment in a bad manner. Though there are also opposite data that an eosinophil might be able to kill directly eosinophils or provide T-cell help. So the first thing that we did is validating results in more patients, not only using the flow data, but really using the data that you get from the diagnostic lab on your differentiation of your white blood cells. And you see that this increase in eosinophils was mainly seen in responding patients, while in non-responding patients, the data was all over the place. And next question that we asked ourselves is that is anti-PD-1 based immunotherapies in metastatic triple-negative breast cancer, Of course, there's a clinical application, there's a certain response rate, I would say between 10-20%. But the problem of acquired resistance is what we observe in the clinic. So patients have an initial response, but after, half a year, 1 year, 2 years, they relapse. Similar, I think, to what is now seen with non-small cell lung cancer, in contrast to this nice plateau in melanoma. And our question was, so is it the case that those women that have a high increase in eosinophils do better? And this plot suggested that might be the case. So here you see it kind of a plateau. Of course, numbers are small, but that patients where you really have a steep increase on eosinophils in the blood, that they do better. Okay, of course, interesting results. But then, of course, the question was, is this like a bystander effect, or are the eosinophils of functionally importance? And that's, I think, where the elegance of such a collaboration comes in, because you can start addressing that question in a mouse model. And we started with depleting the eosinophils using anti-cyclic F. Cyclic F is expressed on eosinophils, also a bit on basophils and mast cells, but to a lesser extent. And we were able to completely deplete the eosinophils using this depletion approach. And what is happening in this mouse model? So again, the chemotherapy, immunotherapy data. But then so in blue, you see the result, but then if you deplete your eosinophils, you affect this durability of response to chemoimmunotherapy, which was particularly profound in mouse models mimicking a metastatic disease. So you have to compare here the blue versus the orange curve. So in the orange curve, the mice select eosinophils. And next we were like, okay, if you now deplete eosinophils, what is happening to your tumor microenvironment? In this case, the tumor-associated CD8 T cells. And we looked at particular characteristics of those T cells. And you see that upon depletion of your eosinophils, you get a drop in interferon gamma and also a drop in CD44. So CD44 is an activation marker, suggesting that the circulating eosinophils do affect the tumor microenvironment. Puzzling results, interesting, of course, more questions than answer. And we try to map all those steps. So what is important to, get those eosinophils kick-started, and what is, subsequently important to change this tumor microenvironment. And here you see data of the plasma of the mice. And we asked the question, what kind of chemo and cytokines do change? You see that if you treat in vivo with checkpoint blockade, you see an increase in certain chemokines and cytokines, but especially on IL-5. And it's known mainly from the field of allergy, asthma, parasitic infections, that IL-5 could be important for eosinophil recruitment. And next, what we did, we blocked IL-5, and here you see indeed that upon blockage of IL-5, that the eosinophils in the blood are lost. We also know from other fields that IL-5 is mainly produced by CD4 cells, and I don't show you all the data, but indeed, if you study indeed the mRNA expression of IL-5, you see an increase of IL-5 in CD4s if you treat with immune checkpoint blockade. And the elegant thing with this, having co-clinical trials, that you can go back and validate it in your patients. So that's what we did. So here you see in responding patients, mRNA expression of IL-5. And indeed, you see an increase after treatment with nivolumab. And also, if you put then the PBMCs, the CD4, put them in culture and treat them in vitro with anti-PD-1, you also see a production of IL-5. So both mouse data and human data suggest that, with checkpoint blockade, we are increasing IL-5 production by CD4s, and that could lead to systemic eosinophil expansion. But as said, in a mouse model, and also what we see in patients, most of the times chemotherapy is needed. So the next question was, what happens if we add the chemotherapy to this profiling of cytokines and chemokines? And here I showed you, with immune checkpoint blockade, the IL-5 goes up. But then if you add the chemotherapy, you see that IL-33 is, increasing. IL-33, and the next step was we did, so is this something that is seen only in the serum or in the plasma, or is this really tumor-based? And indeed, in the mouse models, upon treatment with checkpoint blockade and chemo, you see that there's more IL-33 detectable in the tumor. And next, what we did, we tried to neutralize IL-33. So it's difficult to really block IL-33 in a direct manner, but there's in collaboration with Rudy Beijert, who works in Belgium, in Leuven, there's a kind of IL-33 trap that you can neutralize it at least. And you see here, if you look in the tumor, that on this IL-33 trap, you see a reduction of eosinophils in the tumor. And next, what we try to do is measure eosinophils in the metastatic biopsies, which is very complicated. So you only have one slide, and eosinophils are, even if you stimulate them, are very rare. So, you know, for most patients, we had a feeling that we, you know, difficult to detect, and you highly likely miss them. So here we rely on bulk RNA sequencing data, because then at least you have the, you know, the whole biopsy that you freeze, that you've frozen down, and we looked here at established eosinophil signatures. What is remarkable here, so in responders versus non-responders, that in responders, the IL-33 expression was going hand in hand with the increase in eosinophil, which was not seen in patients not responding to the treatment. So this data suggests that the, you know, additional induction of IL-33 by chemotherapy could promote the intratumoral eosinophils. And now what we are doing, and a bit of it is already published in the paper, is that we see whether with certain approaches we can basically replace the chemotherapy, because chemotherapy is still giving most toxicity and hampering quality of life for most of the patients. So here you see the data that if you treat with recombinant IL-33, and if you look at blood eosinophils, and with checkpoint blockade and recombinant IL-33, we are able to also have this increase in eosinophils. And also in the mouse models, you see that if you leave out the chemotherapy, but combine with recombinant IL-33, that you might be able to, you know, to get the same results. And this is, of course, needs more follow-up, especially on the preclinical side. And interestingly, if you look again at the tumor-based CD8 T cells, what this IL-33 treatment is doing, you see a particular upregulation of PD-1 on CD8 T cells, which might be a very favorable phenomenon, of course. So the paper ends with a model, which again has more questions than answer. But what we propose is that the treatment of checkpoint blockade, you know, induces somehow CD4 T cell activation. The CD4s start producing IL-5. This results in a systemic accumulation of eosinophils, production of IL-33. It's still difficult to dissect, you know, what is producing this IL-33, but at least this seems to be a critical step to get the eosinophils in. And interestingly, you see more and more, you know, movement in this field, I would say. So there was 2 weeks ago in Milan, there was an ACR meeting together with European immunotherapy organizations. And there was this publication, translational study done by a PhD student in the lab of Sylvia Adams, who is in NYU, suggesting that the type of macrophages might be important to recruit these eosinophils. So I think we will learn more in the coming years on this crosstalk between eosinophils, T cells, and maybe even other immune cells. Also, this is not a standalone publication, I would say. So you see more and more papers coming up. For example, this recent paper in non-small cell lung cancer. It's a clinical trial where they did comprehensive analysis. And you see also that blood-based, you know, dynamics of eosinophils seem also to matter in non-small cell lung cancer. Last but not least, a proof of concept trial, basket trial that is ongoing at our institution in the neoadjuvant setting. And we select based on TILs. So it's not particularly that we only select TIL high patients. So we started out with, you know, allowing patients with all TILs, but by stratifying, we were able to make sure that there was an equal distribution, and also in later cohorts select on TIL high tumors. And why are TILs so important in triple negative breast cancer? Of course, you can envision as IO enthusiasts that the TILs do matter for outcome of anti-PD-1, but also they're strongly prognostic. So what you see here is triple negative breast cancer, all under 40, because this was a project started years ago with an interest in young women with breast cancer. And this was selected in cohorts in the 80s and in the 90s. And at that time, the guidelines recommended only chemotherapy for node-positive disease, which is, of course, quite different than what we do right now, where we basically treat all triple-negative breast cancer patients with chemotherapy. And I don't say that's a good thing that in those days we didn't give chemotherapy. At least it allows us to study really the prognostic value of important markers. In this case, TILs. And what you see here at predefined cutoffs, it really matters how your prognosis is. So for the TILs, so patients with low TILs, so you say, oh yeah, we're glad that now we give chemotherapy. But for these women in green, so TILs above 75%, the prognosis is excellent. And you really could ask yourself the question, so is it really necessary to treat those women with 4 types of chemotherapies? Just a sidestep that this, retrospective data, we hope to validate it in a prospective manner. So the prognostic value of TILs, and can we really select a patient population that doesn't need chemotherapy? So chemotherapy optimization, so only treat with surgery and radiation for women with TILs above 50%. And it might be a global trial. We are currently talking to people from ECOG, and maybe it would also be wonderful if SWOG would be involved if we are able to get this from the ground. Okay, that was a sidestep on the importance of TILs in triple-negative breast cancer. But having those data, seeing responses in triple-negative to anti-PD-1, we have the strong hypothesis that we really feel that there's a subgroup of early triple-negative breast cancer patients that is highly sensitive to immunotherapy, and for which in the future, chemotherapy might be de-escalated. So it's a TIL-driven trial. And it's, again, a basket trial with small baskets that we were, flexible in testing our concept and not generating, data in large patients that's a bit more concept-based. So this was the starting point. Okay, start with anti-PD-1. We started at that time, there were no data in the neoadjuvant setting. So we started with a 4-week regimen, and then all patients went to the regular therapy. So this is a window of opportunity design. I will show you later that we left this window design because it has, so much potential that it can really be a neoadjuvant treatment, not only in a window setting. The endpoint, as said, was defined in an era where there were no data in the neoadjuvant setting. So we started kind of carefully. So, okay, let's see with 4 weeks of anti-PD-1 whether we can induce CD8 T cells or interferon gamma gene expression. And as said, so it's stratified based on TILs to make sure that we could study also in, TIL high as well as in TIL low the efficacy of this treatment. And here you see one of the first results we presented last year at ESMO. So this is a waterfall plot based on MRI. And it's only after 4 weeks of treatment. But what you can appreciate that around 25% of the patients after only 4 weeks of treatment already have a partial response on MRI. And in blue, the women that received anti-PD-1, and in green, those with combination treatment. And of course, the numbers are too small to directly compare, but it suggests that if you add anti-CTLA-4, you might be able to induce more responses. Interestingly, you see those NTs, no tumor. So again, I remind you, this is a window setting. So we take a biopsy and then the patients proceed mainly to neoadjuvant chemotherapy. And when we saw this, with the first biopsies dropping in, we had a serious discussion with the radiologist, like, what are you doing? Because all those biopsies are seem to be empty. But it was highly correlated with the results on MRI. And if you see a shrinkage of the tumors, it's highly likely that at least the majority of the tumors might already be gone after 4 weeks. And then you see those stars, and those stars were those stubborn breast cancer patients that were like, okay, I would like to have your immunotherapy, but I'm not going to do the chemotherapy. So we took them to surgery after lots of discussions. And you see, for example, the lady here in pink, that even with only 4 weeks of anti-PD-1 based treatment, she reached a PCR, which was, really a surprise to us, because here you see the results of her MRI. And I think there's not much experience in the neoadjuvant setting with immunotherapy alone, because usually it's combined with chemotherapy. But this is how it can look if you treat with IO alone after 4 weeks. So basically see, if you would do a RECIST measure, that the diameter would be the same, but the aspect of the tumor is totally changed. And we took her to surgery and she has a PCR. So that, I think it shows that, you know, you get an influx of your T cells and the MRI, you know, it's not able to discriminate whether it's a tumor cell or a T cell. So then a look at the biological endpoints. So the pre-specified endpoints that, you know, a cohort could continue if we saw like at least in 5 out of 15 an immune activation, which was really the case for both approaches. Though a bit complicated that we learned. So if you are already high in CD8, it's kind of more difficult to get a doubling of your CD8. Also, if you look at TIL, if your starting point is a TIL of 75%, you will never double your TIL. And thereby those patients are not classified as immune activation based on this endpoint. So at least we learn a lot on window designs and IO related endpoints. We were able to also, look at the ctDNA. We collaborated with Natera, so it's the Signatera assay. And even in this relatively, low stage triple negative, we saw that 80% of the patients were positive on ctDNA. And this is what you see if you look after only 4 weeks. That again, around 25 of the patients with 4 weeks showed a clearance of their ctDNA. And this was highly enriched for responding patients. Last but not least, so some of the biomarker work it's still ongoing, but we presented the first bits and pieces last year at ESMO, and especially intriguing results on spatial analysis without using expensive technologies like Cosmix or Visium or Xenium, so a simple CD8 immunohistochemistry staining. So here you see the patient's radiological response, yes or no. The TIL values as expected, higher TILs in those that responded, similar trend for CD8. Clearly, those with high interferon gamma did better, but here you see the spatial data for CD8. So if you have a low score, it means that the distance between tumor cells and CD8 it's more close. And you see that in patients that responded, that the distance between tumor and CD8 was significantly reduced compared to the patients that didn't show a response. Suggested that not only TIL counts as a starting point for this trial, but really spatial distribution does matter. And now I think it's a challenge. How can we make this information available at baseline and do a biomarker-driven basket based on these findings. And what's now going on is that we kind of, left this window design because we saw, pretty interesting responses, and we now take only lymph node negative patients. We treat it for 6 weeks, and we all take them to surgery. So these were some bits and pieces and flavors on how preclinical and conceptual work can feed, signal finding trials that on their own can inspire large, more practice changing trials. And of course, this is not work done by my own, but this is my research group, really a mixture of immunologists, bioinformaticians, as well as clinical fellows, close collaboration with, the group of Karen de Visser, which is great to work with her and collaborate on the preclinical space. Pathology is key if you want to do those biomarker-driven trials. And, I have the luxury, I would say, to work with heroes in the field who really boost the field of mostly melanoma. And that as, a breast interest working on cold tumor type, we can really learn and collaborate with, with those wonderful people. Thank you so much for your attention, and looking forward to the discussion.
Thank you very much for that amazing and very impressive presentation today. I'd open up the floor if there are any questions, either online or in the room. I have a question that I'd like to start with. I mean, I think the work on the eosinophils shows the importance of being able to look at other cell types and really being able to having to measure these as biomarkers as well, potentially. And you alluded to how that can be challenging. I'm just wondering how you're thinking about that now, like as a next step, like how would you see that moving forward or building it out to be able to detect those cells?
Yeah, so your question is related to the eosinophils in the metastatic where we showed this increase, right?
Yes, correct.
Yeah.
Correct.
Yeah, so this, I think, also from the reviewer side, we got a lot of questions on potential clinical utility. I think, so at least to me, this eosinophil data, I think it would be difficult to use it as a biomarker. Yeah, I might be wrong, but there are in oncology no on-treatment biomarkers yet that feed treatment decisions. So for us, it's more, and also how we are now outlining, future experiments, it's more that it provides mechanistic insights and also opens up avenues how you could, help to get the T cells in, basically, and to get an efficient T cell response. And on the whether to use it in the clinic to make decisions, I think that would be difficult, right? If you have started a treatment and regardless of your CT results, stop or continue a treatment. But I'm of course open for other, visions that and seeing whether on-treatment biopsies can make it to the clinic.
Thank you. I think we have a question in the room here.
Hey there, great presentation. I had a quick question about the Bellini study. I'm interested in your thoughts about a clinical question. I think we all now appreciate, right, that high TILs are a predictive biomarker of immunotherapy responsiveness, and very intriguing data that single agent IO can induce a path CR in that setting, even for a short duration of time. But we also know that high TILs predict chemotherapy sensitivity in triple negative breast cancer. And so I think one of the questions maybe we'll struggle with clinically is that, are these patients who would get path CR just with single agent IO also a group of patients who would get path CR with standard chemotherapy? And you could almost ask the reverse question of, should we be de-escalating the IO? Because from a toxicity perspective, I think there's a debate about, single agent IO versus, ACT chemotherapy and sort of what would be more tolerable. And so just curious about the thoughts there.
Yeah, I think that's a very important topic, especially considering the long-term toxicity. So I think, single agent IO in the short term is usually for most patients fine, but I'm always impressed and, shocked to say if we induce an adrenal gland insufficiency and the long-term consequences for certain patients. Of course, we see also induce thyroid problems, but that's usually, at least how I see the patients, that's okay. But it's adrenal gland problems and other long-term IO toxicity is definitely, have to take it serious. I think the question whether, the neuropathies or heart problems long-term with ACT versus IO toxicity on long-term, it's indeed a question. What and how does it affect the long-term, the quality of life on the long-term? And coming back to this relation with high TILs, I think you're right that maybe for those that do already good, we add IO or we replace chemo with IO, while the patients that are most in need, so the low, maybe intermediate TILs, both of them, whether it's IO or chemo or the combination, it's still not doing a perfect job. And I think it depends on who you talk to, whether, the chemotherapy is considered, more tolerable at the long run, or whether it's single agent IO. I think you need large studies to address that question, and in the biomarker population. I think you had the discussions before that, to enter large patients in a biomarker-driven trial, it will be challenging, but I hope we will get those answers one day.
Thank you very much. So we are past the end of the committee meeting. So I think we're going to close the meeting out now. I'd like to really thank you very much for that fantastic presentation, and all of the speakers today for a great immunotherapy committee session. So thank you very much, and have a great rest of your afternoon. Thank you.
Sure. Thank you so much.